Boa tarde, pessoal. Boa tarde, pessoal que está aqui presente. Queria agradecer a presença de todo mundo e também agradecer ao pessoal que está assistindo de casa. Eu vou apresentar o Dr. Brandon Wilder. Então, como ele é um convidado estrangeiro, eu vou mudar para o inglês para apresentá-lo. Então, it's a pleasure to have Dr. Brandon Wilder. He's an associate professor at the Oregon Health and Science University. He did his PhD at Florida, and he spent a time in the postdoctoral, as a postdoctoral in the Seattle, in the Center of Infectious Disease. And he is not the first time he came to South America. He spent 4 years working in Peru. He was a unit head of the immunology and vaccine development in the Department of Parasitology at the Naval Medical Research Unit in Lima, Peru, between 2019 and 2023. And he works in his lab with mouse models, studying mostly malaria. And he used mice and humanized mice to study vaccines and monoclonal antibodies for therapy against malaria. So it's a great pleasure to have you here.
Thank you so much. Thanks for a nice introduction. And thank you for having me. It's been great. I've always wanted to go to Brazil, and since I was a kid, now my wish has come true. So I'm at Oregon Health and Science University, and our campus is co-located with the Oregon National Primate Research Center, which is one of 7 national primate research centers in the U.S. And this is a picture of it in the winter. It's not usually that snowy, but it's a beautiful large campus. If anyone's ever in the area, please come. We can show you around. We have over 5,000 monkeys there. We do various research from reproductive health, infectious disease, metabolics, a lot of really cool stuff. It's a great place. And before I start talking, I want to make sure I thank our lab, that's small and growing. They're really phenomenal. And like you mentioned, I spent the first 4 years of having my lab in Oregon living in Peru, where so the lab really had to exist and grow and without me, and I think that was probably for the better. That's why it's so successful today. But also, of course, we've been working with Caroline and Luna on a project on malaria, and it's really honestly the most exciting project we have going. I'll mention some other folks as we go along, but what we really try to do is, I guess the thing that I try to do in the lab and with others is kind of take the whole complexity of a parasitic infection, which you are all very familiar with, and plasmodium infection, and think about all of that when we're designing vaccines and trying to understand immunity. And I promise I won't go over the life cycle with you. But this is really just how, we think about infection in terms of the immune system. And, when the parasite is first in your skin, it's really susceptible to antibodies. And that's been a great target, and we'll talk about that more. But then, when it advances into the liver and it's in an hepatocyte, it's a totally new infection. It's intracellular. It's changing. Antibodies are really probably no longer capable of working, and it's the domain of CD8 T cells that can recognize and kill infected hepatocytes. And then when the parasites emerge again, they, of course, change into a new life stage. They look completely different. To the immune system, it's basically a different pathogen, and they infect a new cell type, red blood cells. And again, are newly susceptible to antibodies. And, I think, depending on the parasite, to CD8 T cells when they're inside. And then, of course, all of this takes place not just, at the parasite immune system level, but then the parasite is, of course, as all parasites do, controlling the immune system and modulating it so that you can be infected again and again. And so this is a lot to take into account when thinking of vaccines. But starting with kind of the simplest version of this, and one thing that has really driven malaria vaccine development is, antibodies against the sporozoite. And I love this old videos from Jerry Vanderberg's group showing on the left, this is a mouse ear that was bitten by a Plasmodium infected mosquito. And you can see all of these are sporozoites. And they're all looking for a blood vessel, which this one has found. And then they'll kind of burrow through the blood vessel and then shoot off. And then the infection starts. And on the right, it may look like this video didn't start, but it actually did. And this isn't a mouse that was vaccinated with live attenuated sporozoites. And it has lots of antibodies against the sporozoite, and in particular, the circumsporozoite protein. And so this was kind of the first demonstration that these antibodies, stop the parasite at the very first moment of infection. And this traversal, this mobility in the skin is really important in starting an infection. And that's really driven, a lot of vaccine design. And the two vaccines we have for malaria, for Plasmodium falciparum, are both antibody-based vaccines that target the circumsporozoite protein, CSP. And they're really, infection blocking, and they stop the parasite in the skin. And they're a great start. I mean, they weren't licensed for no reason, but both of them suffer the same flaw in that they're short-acting. And the reason for that is, for all the reasons we talked about before, these antibodies only act at a single stage of one of the three stages of the parasite, and their short activity is related to the fact that you need really high titers of antibodies to protect. And once antibodies wane from your vaccination, as they normally do, they drop below this threshold of protection, and you need to have a booster dose to get those antibodies up high enough to keep protecting. And so, while this protective window offers a lot of protection and will be huge, is, really a big moment for malaria vaccines, it's just kind of the beginning, because you can't afford to keep boosting children every single year, and it's just not a sustainable option. I don't know why I was going there. So again, this is just the start. This is one of over 5,000 proteins the parasite has and targets only one of the three mammalian stages. And so thinking about CSP, they're great in the skin, but the blood stage does not have CSP. The liver stage, there is some of this protein around, but it's probably inaccessible to the antibodies. So that we really want to kind of expand our view into other stages. And one big question that's always plagued the field is, well, what do you need? So the R21, 75% protection against disease for a year. That's great, but apparently not enough. What do you need? And I really like the work of Melissa Penny, who's been doing a lot of modeling, and she incorporates a lot of the complexities of malaria infection and transmission in her models. And I won't go through each of these individually, but she uses all these variables. And the end result is you need, for a vaccine to really eliminate or give long-term control of malaria, and this is Plasmodium falciparum, you need a vaccine that can get into, roughly 85% of people that need it. It has to have a protective efficacy against infection of over 80% and last for over 4 years. And that's a really, really high bar. There aren't even, I don't know many vaccines aside from maybe yellow fever and MMR that achieve that level of protection, let alone against a parasitic disease. And so one idea is to, okay, well, CSP antibodies are great. Can we make just a better one and squeeze more out of CSP? And the traditional path for that is to go through monoclonal antibodies. And CSP has kind of paved the way for that. So there have been two monoclonal antibodies against CSP, CIS43 and L9LS, that have advanced to field trials. And both of them have had really good efficacy. This is both of these are field trials against protection against seasonal malaria. These are the placebo curves of infection, and then you can see both of them have anywhere between 55 and 80% protection against infection for at least the 6 months of transmission, which is phenomenal. I think anybody in the HIV field would probably give their firstborn child to have these results, but unfortunately, it's not enough. And there's two reasons. One is, to achieve this, these were really high doses. These were mostly over 40 micrograms per mil serum concentration, and you can see the doses here, either in milligrams per kilogram or total dose. That's just really expensive right now. Monoclonals, their biggest drawback is they're expensive if you want to use them directly. And kind of going back in some of the pharmacokinetics, it looks like you need roughly 20 to 40 micrograms per mil to mediate this protection. The other limitation is, if you notice in this group, they gave a 4-fold increase in dose, and in this group, a 2-fold increase. And if you look at the protection curves, they really aren't that different. So you're doubling, tripling, quadrupling the antibody dose, and you're not getting much gains out of that increased dosing of an already expensive treatment. And so for us, this really kind of raised a couple questions that we wanted to ask, and those questions are, what are the limits of a CSP-based approach? These monoclonals are the most advanced that we have for CSP and the most advanced for many infectious diseases. Can we do better, and what are the limits? And then the other question is, are we measuring the right thing? Is protection from infection really the end result, and can these be used differently to, think about protection in different terms? And so we rely a lot on preclinical models. These are really questions that you need to sort out preclinically. I think malaria plasmodium is blessed with the fact that, there's many species of plasmodium that infect many species of animals, from chickens all the way to us and everything in between. There's mouse models of plasmodium infection. But what you can see is that the animal models are pretty far from, Vivax is up here, falciparum is down here. The animal models are phylogenetically very distinct. And even within humans, Vivax is a very different parasite than falciparum. And, as you know, the parasites, Vivax and falciparum, don't like to infect our handy mouse models. And so understanding these more complex questions of infection become really difficult in preclinical models. And so for that, for a long time, we've leveraged this humanized liver mouse model. And without going into huge detail, basically these mice are transgenic for a disease where they build up a toxin in their hepatocytes unless you put them on a drug. And so when the mice are born, you have them on a drug. And then once they can move around a little bit, you take the drug away. As their hepatocytes die, you inject human hepatocytes into their spleen. Because hepatocytes in the liver normally regrows after injury, these hepatocytes go into the liver and start filling in the spaces left behind. And you do this over and over again until you have a mouse that has a liver that looks like a normal liver, like normal architecture, but is 95% human hepatocytes. And what this does is means that you can take Plasmodium falciparum, the human malaria parasite, which is normally accustomed to human hepatocytes, put it into these mice, and the parasite will infect and develop fully within these hepatocytes and come out the other side. And so that's great. We've known that for a bit. And so what we've been working, one of the projects in the lab is really using this to understand antibody-mediated protection. And so what we've developed over a number of years is this model where we can take these mice. They have zero immune system, so they're completely immune deficient. You can't vaccinate them, and that's a big drawback. But we can give them monoclonal antibodies or polyclonal antibodies and passive transfer to mimic immunity. And then we take falciparum-infected mosquitoes, and we anesthetize the mice and put them on a cage of falciparum-infected mosquitoes so that they get bit by the mosquitoes and transmit the parasite via the natural route. The parasites will go into the liver, develop, come out the other side, but then they'll come into contact with nothing but mouse red blood cells, which Plasmodium falciparum does not like. So we just simply inject human red blood cells into the mice, and this allows the parasites to infect and replicate and spread. And then we can just take blood for a couple of days to see, okay, did these antibodies stop infection, or did they do anything, to prevent infection? And so this slide, I should say, Peyton is a technician in the lab that's been leading a lot of this work, and she's done a phenomenal job. So any data you see in these mice is mostly her. This is data summarizing hundreds of mice using this monoclonal antibody 317, which is kind of a prototype anti-CSP antibody. We've used this over a dose range, which is shown here, and then over here is the protective efficacy, the sterilizing protection over a number of different repeat studies with the error bars shown here. So what you can see is you increase dose right about 50 micrograms per mouse, which is roughly 20 to 60 micrograms per mil. You kind of cross this threshold of 50% coin flip chance of protection. And then as you increase from there, you quickly get into roughly 80% sterilizing protection. The interesting thing is as you triple and then double the dose again, so you go from 100 to 600 micrograms per mouse, you don't see efficacy increase anymore, and you certainly don't get complete sterilizing protection all the time. And so, the two things to take away, I guess, first down here is the same data, just grouped using the serum concentration of antibodies instead of dose. So grouping them by quartile, and it's roughly the same data. One is somewhere around this 20 to 50-ish micrograms per mil. We see that's where the protective threshold of the antibodies is, which lines nicely with the human data. But there seems to be a threshold effect. You don't get more protection out of more antibody for CSP, and that's was an interesting finding for us, and also backed by the clinical data. But the more interesting thing, I think, is sterile protection is not the end of what you want. And so what we're looking at in these graphs is emerging parasitemia. So the mice that got antibody but were still infected, how many parasites came out of the liver? And so if you look at mice that do not get antibody, we see roughly 10 to the 5th parasites per mil over a wide range. And then the doses down here, the mice that did get infected, the breakthrough infections, you can see starting at this lowest dose, which is only half protective, you see a 2-log reduction in the number of parasites emerging. And then you see slight gains with the larger doses, but again, there's a bit of a plateau here. And this is the same if you break it down by quartile. But the end result is that, if you take all breakthrough mice together, you can get, a 2 to 4 log reduction in the parasite burden coming out of the liver, the one that's establishing the blood stage. And so this led us to believe that maybe for the first time we could answer a long question in Plasmodium, which is, can you combine multiple stage interventions or multiple stage vaccines to get a better result? And blood stage vaccines have struggled in malaria for a long time because they're not good enough. They just do a little bit. But, is this lower amount of parasites now an easier target for a blood stage vaccine? And can they work together? And so for that, we went to the protein Rh5. We were working with the group in Oxford, Simon Draper's group. We simply wanted to ask, and so Rh5 is an essential invasion protein at the blood stage that's advanced clinically. It's essential for the parasite to invade a red blood cell. And we wanted to ask the question, okay, can you combine this stage plus this stage and get better infection? And we liked Rh5 because it had a lot of good preclinical data, and just recently this year they did a phase 2 trial in Burkina Faso and showed that in infants, it gave 54% protection against disease, which is great. And more interestingly, in these graphs here, this is kind of showing efficacy against any parasitemia and then increasing parasitemia. The vaccine works best against really high parasitemia. So what it's doing is just reducing the amount of parasites. And so the thought is if you start with a low amount of parasites, can an Rh5 component bring that to 0 or bring that to near 0? To do that, we needed to modify our model a little bit. If you remember, we really only monitored blood stage for a couple of days. And so what we wanted to capture was, multiple rounds of infection replication and reinfection. And so we do the same thing that we did before. And instead of just following for 2 days and giving a couple injections of human red blood cells, we followed for about a week, day 7, 9, 11, and 13 after infection, to give us multiple rounds of infection at the blood stage. And if you're thinking that this is a really contrived and probably artificial model, you're not wrong. This is, and so we wanted to say, okay, this is a very weird way of doing things. Does it mean anything? And to do that, the Oxford group helped us answer that question. So they had done a controlled human infection study with Rh5, where they vaccinated people and gave them a blood stage challenge. And this is what their parasitemia looks like. The vaccinees are in pink, and then the controls are here. And what they saw was just a very slight shift in parasite replication. Not great protection, but just like a little bit. And so we took polyclonal IgG from these vaccinees, put it into our mice to try and recapitulate the same level of antibodies that the humans had, and then put it through this model, this challenge. And what we saw is this is what the parasitemia looks like in the controls in black. And then this is what parasitemia looks like in mice that got antibodies from vaccinees. So a very subtle shift. It is a statistically lower area under the curve, lower parasitemia burden, lower days of infection, if not just subtle. So pretty similar to what we saw in humans. So, this gave us a little bit of confidence that we might not be too far off the mark with this very artificial model. And so then we went to do a test, and we went back to monoclonal antibodies because they're easier to work with, and the Draper group had been making really good monoclonals against Rh5. And so these are the results. So basically what we did is on this graph, again, we're showing parasitemia on the y-axis with the limit of detection in red here. And these are the four groups: a control group, a group that got Rh5 antibodies alone, a group that got CSP antibodies alone, or a group that gets both of them together. And the different colors are the different days, day 7, 9, 11, and 13. Each dot is a mouse with like lines connecting each mouse through the course of infection. And so control mice, you can see they have this parasitemia that just kind of persists and then slowly fades over the week. If you give these animals an anti-Rh5, the blood stage antibody, you can see that their parasites start lower, but no mice are sterilely protected. They all get infected, but the course of infection is much shorter. The drop-off is shorter, the area under the curve is shorter, and then they all clear by about day 11. In contrast, mice that only get CSP, you can see roughly exactly half of them are protected from infection. They're sterilely protected. And those that break through, again, start with about 2 logs lower parasites. But instead of going away like the Rh5 group, they just kind of persist, which makes sense because there's no CSP at this stage. The CSP antibodies shouldn't be doing anything, and they're not. But if you put the two together, what you see is that instead of roughly 50% of mice being protected, 80% are sterilely protected. They never show signs of infection. And the few that broke through, the 3 animals that did break through, have a single day of extremely low parasitemia that's clear, that's only present for 1 day. And so I guess I should say, the statistics on this are you don't have to know statistics to say that if this were a human, you want to be in this group. But in terms of sterile protection, not statistically significant, but just off the mark. But every other measure of the resulting parasite, the blood stage infection, are much, much lower in this group, and that's pretty obvious. And kind of thinking about this in terms of if this were to go to a human, for falciparum, if you can keep the parasitemia below the symptomatic threshold and below a threshold that can be transmitted, that's all the things you need for sterile protection. So that's functionally sterile protection. And as you might know, for Plasmodium, in order for it to be transmitted, you need an infection to last for at least 8 days. And so even if you can just shorten the infection to less than 8 days, that infection will stay with that person. It can't be transmitted to mosquito. And so, we think that this is, this is kind of the first evidence, the first data evidence that combining these 2 stages can have outsized effects on a vaccine and may have bigger effects on disease and transmission. So to summarize the first part, anti-CSP antibodies are great, but they probably have limits. I forgot to say one caveat. You'll notice these doses are very different. We give an extremely high dose of the Rh5 and a low dose. And this was really trying to make this experiment work for us. We really wanted to go for just proof of concept and not, is this going to be a therapy for humans? But so we chose a dose of CSP that would give us 50% protection and a dose of Rh5 that we were certain was going to protect because each of these mice costs $2,800. And so we didn't want to have to repeat this experiment because it was very, very expensive. And so next, the kind of big question is, what does this mean in humans? And, can we measure this effect in vaccine trials? And does this hold true for a human getting infected and a human that has an immune system? And then I think another interesting question is, how does this actually interact with natural immunity? Kids get infected, over and over. So the vaccine might have an opportunity to work in conjunction with a multi-stage, a dual-stage vaccine. And then next, another question is, okay, so that's 2 stages. There's 3 stages, and each stage of infection has multiple steps. So how far can we push this? Can we just keep targeting different little parts of the parasite and getting bigger gains? Maybe I'll stop there for questions on that part, if anyone...
Okay. I remember I thought they have a lot of expectation on RH5 because it was not variant. And but your result says that I think you showed that immunization doesn't really protect.
It does. It didn't protect in this, the CHMI.
Yeah.
But it did protect in Burkina Faso in infants. And so the big difference is, I think it comes down to the adjuvant matrix M works really well in infants and not as well in adults. And these infants had much higher antibody titers than the adults. And RH5 is, 1-to-1, really dependent on antibody titers.
So, because I can't see, right, what's the...
So this is survival curves for...
different colors.
This is the placebo group here, and then these are the vaccine groups. Right. And this is protection against, I believe, any infection. And then this is increasing parasitemia threshold. So the most extreme here is like protection against very high parasitemia. Not many kids got very high parasitemia, but RH5 protected completely against very high parasitemia. And so the way I think of it in my head is the vaccine just kind of drags parasitemia down. And so...
so it doesn't give you sterile protection, but it really brings down the parasitemia and the pathogens.
Exactly, yeah. Yeah. With the result that it's a 50% reduction in clinical disease.
And then, but you didn't do relapses on your experiment, or?
No, this is falciparum, but we'll get to the relapses later.
That's right.
Yeah.
This is falciparum. Okay, sorry.
Yeah, used to people not thinking about relapses.
I also would like to ask, have you tried therapeutic use of this RH5?
No, I don't think they have either. No.
Do you think it could work?
Probably not.
Okay.
Another question. Of course, the amount of antibody you have to inject is very high. But do you know how much high do they stay in the circulation after you inject? Because probably a big part of these antibodies are getting sequestrated, so you don't have all of this amount available to inhibit the invasion of the parasite.
Yeah. With the best monoclonal antibodies, you can expect between 1 and 5 micrograms per mil for 14 to 18 months. And so that's kind of where, we're setting our sights is, and you can kind of see this, that's this lower end here, where you're getting very little sterilizing protection, but still a huge reduction in emerging parasitemia. And so the experiments we're doing now are bringing the doses of both the CSP and the RH5 antibodies down to this real world level and saying, can they do anything when neither antibody is at its best, but does this make for, a scenario where they can work at these real world concentrations? And if so, that'll really help, not just monoclonals, but vaccines as well. Because, vaccines, you can get 1 microgram per mil for years and years. So, yeah.
So I would expect that you need much less anti-CSP antibody than the H5 because you have much less parasites, sporozoites than...
Yeah. And that's a part of the other project. We're the end use of this, and the Oxford group is doing a good job of... RH5 is just one of the proteins in this PC-RCR complex. And so we're trying to see in the mice if we can combine antibodies from all of these to get more out of the blood stage, because that's the harder part for efficacy, yeah. Cool. Any questions in the back? Go for it.
My question is about the model. And after you humanize the liver, are the mice back in the drug?
Are they?
They were taking a drug to inhibit the toxin, right?
Yep.
So after it is humanized, they are taking the drug again.
Yeah. Yes, but less often.
Yeah. Okay.
Yeah. Usually Yeah.
Yeah. So my question is about that. how can you be sure like the drug or the toxin is not interfering with your results?
So the short answer is they're off drug for a long time before we do any of the experiments, and the drugs have a really short half-life. So they have no drug in their system whatsoever. Yeah.
Thank you.
So I'm going to go through this next part super quick. If anybody has questions and want to go back to it, we can. That is to say, the first part of infection was oversimplified, and the journey from the skin to the liver, the parasite has to sense multiple environments and use many different proteins to know it's in the skin, it's in the blood, it's in the liver. It needs to choose an hepatocyte. And those are all different proteins and other opportunities to interfere with the infection process. And I won't talk about it because it's been published. We've shown that that CSP has this threshold effect. And if you layer on antibodies against a different protein, SSP2, you can overcome this threshold effect by targeting a different parasite protein that acts in a different way. And so, this isn't unprecedented, and I think, just we need to understand how far we can go. And then I won't talk about it. I have secret slides at the end if anybody's interested. We do have some evidence that antibodies can function against the liver stages, the intracellular liver stages, and this has been published a long time ago by somebody else, and we verified it. And so, again, thinking about, trying to hit the parasite because it has so many steps of infection, trying to hit as many as possible, we're starting to think that that's a good strategy. And then what about Vivax? The models are definitely underdeveloped for this, but we've used this humanized mouse model to do a couple of Vivax studies, one with Chris King's group, using an antibody against AMA1, which is usually thought of as just a blood stage antigen, but it's also functional at the sporozoite stage. And they made monoclonals to a highly conserved region of it that looked good in vitro. And so we were able to give mice this antibody and then challenge them with Vivax sporozoites. These were field isolates from Thailand. And this is showing the liver burden at day 8. And you can see that a high dose of this antibody was able to prevent a lot of the liver stage infection. Roughly a little over 90% of liver burden was reduced. And then this was a pretty strong dose effect. And so while this antibody isn't great, I think it's an interesting proof of concept and probably holds more potential as a dual stage antibody because we didn't let them go to blood stage. So we're excited to try and push this into some of the monkey models that I'll talk about in a minute. So this is great, and to summarize, I think the argument for using multiple antigens that target multiple life stages is pretty strong, but that raises more questions, and some of you are thinking about this as well here, and how do you make a vaccine that targets 3 completely independent antigens? Making a vaccine with one antigen is hard enough. You have issues of what platform are you going to use? Will you get interference between the antigens? Do you need one antigen to have more antibodies than the other? And how do you modulate that? And then the question is, how do you test that? There aren't many models. The humanized mice, they do not have an immune system, so you cannot vaccinate them. And then, thinking long term, how does this impact durability? The goal is to get a vaccine that lasts for 4, 5, 6 years. What are the limits, like I was talking about, when you have very low levels of all antibodies? Can these antibodies work together at the bottom of your antibody titer and not just at the top? And so I think an easy answer for multivalent vaccines is mRNA. But then a lot of these questions become very difficult, I think, to answer in mice, and usually that depends on the antigen as well. And I think one good example of using mRNA to its fullest extent is this influenza paper using 21 different variants of hemagglutinin. So you have a 21-valent vaccine. But then I think one of the interesting things from mRNA vaccines, as we're all very well aware of, is that if you look at antibody titers in humans to COVID vaccines, you get a nice peak, and then those antibody titers wane, and they seem to work less good at this tail, similar to the CSP vaccines. And the problem is that this wasn't really predicted in mice. Mice don't often show this, waning immune response. This is the same vaccine given to mice, and you can see from day 21 to 233, the antibody titers persist. They don't drop off. And so if we want to answer these questions, you have to do something different. So it was mentioned that I was working at NAMRU down in Peru, and we have had the opportunity to work with a non-human primate species and some really talented people. This is the team that worked with us on the studies. There were us parasitologists, there were people that were animal technicians, and then there were entomologists, so we could do vector-borne diseases. We got to work with these cute little guys. So these are Aotus owl monkeys. They're native to Peru and Brazil and the Amazon. And there's a colony of them in Iquitos in Peru, and they're great. They're small, they're easy to work with, they're available, they're relatively inexpensive. They're actually cheaper than our mice, our humanized mice. And they're susceptible to a wide range of human infections, including Zika, dengue, some alpha viruses, Shigella, E. coli. And then you can challenge them directly with the blood stages of Plasmodium falciparum. Hopefully we'll have a paper out soon that you can do the same thing with Vivax. And they have a lot of human immunology conserved as well. And they've been really important in malaria for the Rh5-based vaccines, for blood stage vaccines. And they were the first model to show that, Rh5 vaccines these are parasitemia curves from these monkeys. If you infect them with Plasmodium falciparum, this is what the infection profile looks like. It's a really strong, robust, and pretty severe infection. And depending on the vaccine platform you use, you could get anywhere from high levels of protection with Rh5 with a vaccine regimen in CFA that gives you lots of antibodies, but lower antibody titer regimens, didn't sterilely protect, but kind of gave you this partial protection. And this exactly predicted what was seen in humans. So it's really nice that you have a nice model for falciparum. Let me skip through this. So we had the opportunity to test a vaccine that was both an LNP mRNA and then the same on a nanoparticle in adjuvant, side by side in these monkeys. And these are the antibody titers shown here after 3 monthly immunizations. And just like humans, you see, nothing after the first, a little bit of rise after the second, and then, peak after the third. And these wane on the same 30 to 50 day time period that humans do. And this kind of stabilizes in the normal trajectory, and then they're boosted by an immunization later, just like this looks very similar to what you see for humans. And so this is really nice in that we think that these monkeys can not only be infected with human pathogens, but replicate some of the nuances, the details of human immunology after vaccination. And at least for malaria, one of the hallmarks is you get repeatedly infected, and it takes a long time to develop immunity, and you eventually develop some semi-immunity against disease, but not infection. And we see that in these monkeys too. So this is the parasitemia curve for a first infection, where every monkey reaches high parasitemia and has to be treated. And then these are the parasitemia curves for a second infection, where all of the monkeys became infected, but only half of them, or I guess, half of them resolved infection asymptomatically, and then the other half had to be treated for anemia or low platelets, other clinical symptoms. And so this is a really nice way to measure semi-immunity, how hybrid immunity or previous infection, natural immunity might interact with vaccines. And we see boosting to natural immunogens like MSP1. This is their antibody titers after the first infection, and then as they were challenged, those antibody titers were boosted, so they're responding in an adaptive way. And so this is kind of putting this all together. We've got some ongoing projects, one funded by Open Philanthropy, and to make complicated models even more complicated, we're kind of mashing them together into this general strategy. And so what we do now is we take our non-human primates and we take advantage of the fact that their immune systems are more like us, that they respond to vaccines and can discriminate these smaller differences in vaccines a little better, and use them as our main vaccinees. And then we can, watch antibody titers over a prime after a boost and, look for different features of interference or anything. If it's in Aotus, the little owl monkeys, we can challenge them directly with blood stages of Plasmodium falciparum and then re-challenge them. But if we want to do a multi-stage vaccine, we can do what we did for the clinical trials and take the IgG from these different time points, put it into our humanized mice, and then challenge them with Plasmodium falciparum infected mosquitoes, and get this multi-stage effect, and see if combining different antigens that we've optimized in the monkeys actually does anything for protection against the natural route of infection. And then we also pair this with functional in vitro assays with these different partners around the world for a very complicated, but we hope very human relevant way of testing malaria vaccines. And as one of our first objectives is, again, if you're thinking this is very artificial, we thought the same thing. So we're taking, vaccines that have a lot of human data and putting them back into the monkeys and putting them through all of these to understand how each of these assays actually predicts what's happened in humans. And we're doing this with R21 matrix M, RH5 in matrix M, PFS230 in matrix M, and then combinations of these as well to know, what do and what do we not predict with the monkeys, and how best can we use them. And so for that second part, I think we make the argument that non-human primates are a little better for some of these nuances. Of course, they'll never replace mice in terms of proof of concept, high throughput testing, and we'll find out what they do predict. I think some cool things that we're looking to investigate now are, can natural immunity and vaccine-induced immunity work together? Hybrid immunity, and should we include antigens that may not be great but at least can be boosted with breakthrough infection? One of the nice things about CSP and RH5 is you don't make antibodies to them during an infection, and that means that they're not subject to evolution and they aren't extremely variable. But that means that your vaccine and your own immunity are working side by side. And a breakthrough infection doesn't boost your vaccine immunity. And so can we include components that actually work together? And that's one thing we're really interested in finding out. And then we haven't even talked about CD8 T cells, which are my first true love. Okay, I'll pause there again for questions on the last part. Behind you.
I have a question. Because of the models that you are explaining, you only use mosquito bites in mice, right? You don't use them in non-human primates.
For the non-human primates I've shown thus far, we're only doing direct to blood stage challenge. And
yes.
So that totally avoids the skin and liver stage. Like the parasite, the sporozoites aren't even involved. And so
that's exactly like that's the reason for that, or it's just a strategy?
Yeah, the reason is because the blood stages aren't transmitted by mosquitoes.
No,
so,
yes, yes,
so
of course. But I mean, like, why you don't try the mosquito bites and on human primates?
We did. That was my main project in Peru, and we failed to get a mosquito bite challenge model to work.
Okay.
But for the next stuff I'll show, we're moving to mosquito bites in other non-human primate models.
Oh, okay. That's,
yeah. But I'm glad you think it's important, because I think it's important as well. And sometimes it's a battle to convince people that it's very important. Yeah, it's like critical.
For the multi-stage vaccine, have you done any experiment? And then the question, I think like we have this problem with dengue, which is a big problem because of but for malaria you don't have that problem, right? You may have a competition of an antigen with the immunogenicity, but you don't have a problem of the antibody
enhancement.
Yeah, that's right. So maybe I don't know. You seem to be disencouraged for some problem of different levels of antibody. Maybe it's not that huge of a problem. I don't know.
Yeah, there's not great data on it. So we're a small part of it. This huge consortium. We're like really just the monkey people. And before the vaccines get to us, they go through mice, through rats, and then to us. And a lot of the interference questions are being addressed in mice and rats. And this data is not ours, but it's totally fine to share. Is we don't see huge effects of interference, but when we do, we thought CSP would kind of quiet down the other responses. It would be immune dominant. We actually see the opposite, that CSP performs worse when it's paired with other antigens. And we don't understand why, we want to be cautious that this might be a mouse phenomenon. So our first experiments in monkeys, aside from taking human vaccines in, is taking the vaccines that we've put through these mouse models and those same exact designs into monkeys to see, do we see the same thing? If we do, that's great. We can avoid using monkeys for those questions. If we don't, then we know the limits of each animal model. But I am highly encouraged by the multi-stage vaccine. Cool. All right. So CD8 T cells, and this is really what's brought me here, thanks to Luna and Caroline. So again, we've been talking about antibodies a lot, but CD8 T cells, they've been shown in at least mice and to some degree humans to be really effective at clearing out liver stages. And of course, by Caroline in a really seminal paper about Vivax, showing that CD8 T cells can kill Vivax-infected reticulocytes. But traditionally, you're really thought of at the liver. And we kind of focus on the liver because every Plasmodium species has to go through the liver. And if you can target them there with CD8 T cells, you have a strategy that can be applied across multiple species. But there are many things that we don't know about CD8 T cell immunity that we need to know to make a vaccine, including, what are the epitopes presented and how. And thanks to Luna, we now know. But then to make a vaccine that targets the liver, we need to know what kind of CD8 T cell you want out of it. CD8 T cells are very diverse, and then especially those that are in tissues are very different. And then once we know these things, is there a vaccine platform we can actually use to get these types of T cells to the liver, to these antigens? And, these two papers kind of summarize a lot of what we know about CD8 T cells in the liver. The only previously identified real antigen was RPL6, is this ribosomal protein. And if you make CD8 T cells against this in the liver of mice, you can get high levels of protection. This is 70% protection. And that's better than, these kind of classic CD8 antigens that were more or less, they're just guesses, which give very low levels of protection at using the same immunization strategy. And so I think that's the cool thing, and that work that Caroline and Luna have been doing. Now that we do know the antigens, they're really this weird class of ribosomal proteins, and not what we would expect. And then what does a liver CD8 look like? Everything we know about CD8 T cells in the liver comes from mice because we can get their livers and look at them. And anytime there have been live attenuated vaccines that target the liver in humans. But it's been difficult because the cells, they don't really see CD8 T cell responses in the blood of these people, even though we know we're getting them. And that answer was, the answer of why was provided by this study in monkeys where they gave these live attenuated vaccines, these parasites that go into the liver and then die and really drive a liver-specific immune response. And if you look in the blood of these monkeys for CD8 T cell responses, either interferon gamma or TNF alpha, sometimes you see the responses. When you do, they're low and inconsistent. But if you go and you look in their livers, you can see the immune response in every single monkey that's very robust. So you're really missing, by looking in the blood, you're missing what's happening in the liver. And so, so we know some epitopes, but they're unexpected and interesting. To know what the liver CD8 looks like, we need to go into the liver to look at it. And then all of that will feed into, hopefully we can come up with a platform that can drive these. And so we've really been using the non-human primates. We've gone back to the non-human primates to try and answer some of these questions, given that their immunology is close to us and we can access their livers. And one of the big things is, if you're squeamish at all, I'm going to show a surgery video. We've been working with the Primate Center to develop these surgery techniques. And so what you're seeing is a laparoscopic, a minimally invasive surgery inside the abdomen of a monkey. And this is their liver, and this is called a tissue sealer, and it is really hot. It cauterizes, seals the blood vessels as it cuts, so that you can really quickly go in and take a big sample of liver. And it'll kind of pause here. You can get this big intact piece of liver, and from that, you can get really nice tissue, really loads, tens of millions of immune cells. But then you've got to get this giant piece of liver out of your surgery hole, which is only 1 centimeter long. And that's where this $400 plastic bag comes in. you bring it in, and then put it in this bag, and then you'll see he kind of like pulls it out, and that keeps the piece of liver intact, but allows you to squeeze it out through that little laparoscopy hole up there. And this is in comparison to normal biopsies, which are really just, they kind of like grab and tear tissue away, and you get really bad histology, really low cell viability. And with these, we've really been able to get a lot of answers out of the liver, more answers than before. Okay, so I'm going to go through like the next section very quickly and try and leave time for questions. We use these monkeys in this biopsy to test whole parasite vaccines that drive liver-specific responses. And I won't go into details, but we can vaccinate multiple times, and then we can take up to 6 of these biopsies. And so that allows us to watch the development of immunity in the liver through time. And the upshot of that is, if we do this vaccination and take biopsies, we look in the blood, we look at the CD8 responses after 1 vaccination or 3, and in the blood you don't see any boosting to any of these kind of normal antigens. We do to Luna and carolinase antigens, but if you instead go into the liver, now between the 1st and 3rd vaccination, we see a boosting. So we're actually seeing the development of immunity by doing these biopsies, and they're worth the time. And we've really been pushing this to the limit by with the help of these 3 guys, by doing these complicated studies, looking at previously infected monkeys, naive monkeys, and then pairing with all sorts of crazy, expensive bioinformatics and single-cell sequencing. And it's really nice. We can get partial protection. We can get 50% of monkeys protected. So we can look at what immune responses correlate with protection and which do not in the tissues. And I'm going to skip this. If anybody cares about NK cells and the type of correlates of protection, we can talk about it. I'm sorry. But I want to talk about Vivax. You all know that Vivax is really hard to work with. You have to wait for somebody to get infected, to show up sick, to get their blood, to get them to consent to give a bunch of blood before you give them treatment for the disease they're seeking treatment. And it's tough, and you have to do this in places like Porto Velho or Iquitos, where we were, which is not ideal. It's I don't have to explain to you how tough it is. And to overcome that, we've been using Plasmodium cynomolgi, which is a primate malaria that's extremely closely related to Vivax and replicates Vivax infection in terms of hypnozoites, relapse, and reticulocyte restriction at the blood. And so we really like it. But it doesn't always match in the antigens, specifically for CSP. The repeats are very different. So to overcome that, we stole a strategy from the mouse world, and we took a Plasmodium cynomolgi parasite that can be cultured and used CRISPR gene editing to swap out to replace the cynomolgi CSP with a Vivax CSP that expresses all 3 of the serotype variants. And then we took this transgenic parasite into monkeys, expanded it. We saw that it gave a blood stage infection, which was a relief. Transferred that to a new monkey, fed that monkey's blood after it was infected to mosquitoes. We got sporozoites. Those sporozoites went into a new monkey. We saw a primary infection after sporozoite infection. You clear that infection and then wait, and then that monkey then relapsed, showing that it has hypnozoites and has the capability of relapsing. And if you look at the sporozoites, if you look at antibodies against either the 210 variant or the 247, isolates from either Thailand or Peru only show up green for the 210 variant and not the 247. But our sporozoites here from our transgenic are recognized by antibodies to both the 210 and the 247. So they actually are expressing the protein, they can infect, and they can relapse. And so I think right now we're moving to mosquito bite infection using this parasite, and we're really excited to put it to use, potentially with one of your vaccines. I'll stop there to leave time for questions. Thanks for listening. I'm sorry if I glossed over quite a bit, but I appreciate the questions. Thanks.
Questions?
Ribosomal protein the same as the caroline, or they're different?
Different, yeah. I mean, the same in that it's a ribosomal protein.
Right, but it's
and
the same, different protein.
yeah.
There is no any similarity. No.
No, I mean not sequence-wise. I mean, yeah, you would know better, but I don't think so.
The RPO6 is different, but the pool 9 that he showed is the same difference, ribosomal. The pool 9 that he showed that induced the response in the liver stage. Did you see the pool 9?
What's pool 9?
It's 9 ribosomal protein that you find in the immunopeptidomics.
And that's
Yes, it's
Yeah.
the same of the we are working. Yeah, there, 9
this one here.
ribosomal proteins.
Yeah, these are
That
all
we
Luna and
found.
Carolina's,
very good in boosting.
Yeah.
and across species, or conserved across species.
Oh, yeah, that's right.
Yeah, I'm finding very interesting this data because you don't see any induction of immunity if you look at the blood.
Mhm.
But then in the liver, have you characterized these T cells to see if they are like peripheral?
Yes, and that's like a part of this study where we vaccinate monkeys and we take many biopsies of the liver, but also the spleen and everything. I mean, this generates a massive amount of data. We have tens of millions of single-cell-seq pieces of data. The CD8 T cells seem to be the least interesting in the liver, which is weird. But all the other supporting cells seem to be very interesting. NK cells are a big indicator of protection. Gamma delta T cells seem to be responding more robustly. And that may be a way we're analyzing the data. We're just missing the activation of the CD8 T cells. But going forward, we're going to try, now that we have antigens for CD8 T cells, which we never had before, we want to go back and try to characterize more specifically the antigen-specific CD8s. But we just didn't have enough data or enough sample to do that in these.
But if you think that you are moving these candidates for a clinical trial, for example, you will need a biomarker of immunogenicity. How do you plan to solve that?
Yeah, I mean, I guess if we're going to target the liver, I guess there's not a vaccine platform that can do that yet. And the only ways to get liver targeted responses now is to intravenously inject things, which is fine for adults, but not great for children. I guess the hope would be that you would give a vaccine that could be administered peripherally in the muscle, and then part of that vaccine would be targeted to the liver to drive a liver-specific response, either if it's a targeted LNP or some viral vector that can spread to both. And the hope is that the peripheral component would give you some immune response peripherally as well as in the liver, and so you might be able to capture both. But yeah, I mean, yeah, it's tough. Ideally, it just works so well that you can go off of efficacy.
Yeah,
There was all this discussion that if you give IV, it goes much more to the liver.
Yeah,
That was the sporozoite experiment, right? With,
exactly. Yeah. Yep.
Steve Hoffman.
Yep. Yeah.
And
Yeah.
Bob said.
And they can vaccinate babies IV, but yeah. I think infants are fairly easy to vaccinate IV. It's like, if anybody that has children, when they get 2 to 5 years old, it'll be a little harder to sit them down and get them.
Yeah, I really like the last slide that you showed this difference in the previous one when you showed the difference between the T cell response in the blood compared to the liver, because this is something that I always said, that we are when you're doing immunology in humans, we are only seeing half of the story, because you're only looking at the PBMC, and sometimes the blood doesn't match what you found in the tissue. And my question would be whether you see differences between these cells and in the expression of the receptor chemokines. But
yeah, it's huge. I mean, that's one thing that's really slowed us down in our single cell sequencing analysis is you go in the liver and you try to identify activation, memory commitment, anything. And what you're going off of is either mouse data or human data from PBMCs and maybe human peripheral blood that's been showered with peptide or SEB or something in a petri dish. And none of those rules apply to actual cells within tissues. By using traditional ex vivo markers of stimulation, we see no activation. So we've had to go back and reverse engineer. Because we have these longitudinal time points, we know our interventions, and we can say, look, these monkeys got vaccinated 2 days ago, their livers are full of parasites, what's different about them from baseline, and use that signature as a real activation score. And when we do that, we get a very clear picture of activation, but it's not your typical interferon gamma TNF alpha. It's a lot of other commitment markers. It's completely different. So it's fascinating, but also frustrating at the same time.
And what would you say is a big pitfall or limitation working with non-human primates?
Yeah, I mean, there's a lot. We always say that their immune system is closer to humans, but we don't know. Because they're very expensive and they're, smart, you don't want to overuse them. And so a lot of people use them, a lot of regulatory agencies require them or like to see them just done, and they are used as a stepping stone. You're like, okay, it worked in mice, it works in monkeys, let's move to humans. And you miss a lot of the really deep immunology that you could get out of them to answer these really deep questions. And so because of that, there haven't been tons of tools to do this, and that's frustrating and a limitation. The other is, they're just different than humans in a lot of ways. There's certain immune subsets that aren't the same in monkeys and humans. Diseases don't always overlap. And yeah, that's about the biggest drawback I can see. Otherwise, I love them. They're great.
Thank you. Any more questions?
I have a question about the evaluation of the load in the liver, because you do it by PCR, right? Because we don't have another techniques. But I think that is like probably the protection, I don't know if I can say like that, but it's probably bigger than you are actually seeing, because when you are looking to the PCR, you have like schizogony, and then the load of the 18S will be really, really high. Yeah. So I don't know, did you guys are working to try another strategy to evaluate this load of parasite in the liver, because we know that is really, really difficult. Yeah. So it's just like a question of interest to see if you guys are developing something to overcome this.
Yeah, 100%. I think, where we can, just using the development of blood stage and that infection is the best measure I think we can use. But with Vivax, that's more difficult. So then we will more heavily rely on the cynomolgi model in monkeys. We haven't gone in to kind of biopsy the monkey livers to assess that directly, but we want to. The only other technical way that we've done it in that study, I didn't show it, but we took those samples at day 8, which for Vivax is like the end of the liver stage, but also the beginning of the blood stage. And in the untreated monkeys, we could detect Plasmodium proteins in the blood. So if you use an ELISA for Vivax proteins, we got positive responses. If we did that for the antibody-treated group, even though we saw liver infection, we saw no blood stage antigens around. So kind of like a rapid diagnostic test or an antigen-based test. So I think that's a viable route. We just need to try and use that a little better. And I think that gets it, if there's non-viable parasites in the liver as well, that might help us out.
Yeah, and I was thinking also about, because you get a really huge piece of liver. And I don't know, maybe it's a really, really dream to see a immunofluorescence, for example, but I know that it's really, really low parasitemia in the liver, but I don't know if there is a way to overcome this.
It's my dream as well. And I think so one thing I didn't show is we have little squirrel monkeys, and they can be infected with Vivax sporozoites. And so we just did a study where we infected them with Vivax sporozoites and took a biopsy of their liver. and we have yet to do the immunofluorescence, but my hope is that we can see Schizons, hypnozoites, and these are immune-competent animals, so we can, for the first time, see immune cells around there. And so we'll do like spatial transcriptomics.
That would be amazing.
I know, I'm so excited.
Brandon, I have to go, but great to have you here. Fantastic. And hope you come back. I think you're leaving soon, right?
Tomorrow.
That was next time you stay longer.
I hope so, yeah. And thanks for having me again, and thanks everyone for taking.
Thank
so
you
much
very
time
much.
today.